I'm Anita Bhattacharya, and I'm a researcher at the Weissman Center at the University of Wisconsin-Madison. And my lab uses human stem cells to study human brain development. So I got interested in Fragile X when I started working at the Weissman Center. I was really impressed by the breadth of research that was going on there, from clinics seeing patients and individuals with different developmental disorders, including Fragile X, and then my end of the research, which was studying the very basic research, basic developmental processes in Fragile X brain development. Well, I think for basic researchers, one attraction of Fragile X is that we don't really know how our brains develop. Most of what we know about how our brains develop is work we have that people have done in animal models. And so when you have a disorder like Fragile X, where there are specific things that are known to be going wrong, it provides you with a way to not only try and fix those problems, but also to learn, oh, this is how the human brain develops, and this is, at this point, something here goes wrong, and this leads to something like Fragile X and the symptoms of Fragile X. So I think it is attractive not only in that we would like to help these individuals, but also as a way of learning more about normal brain development and how it really works. My lab has two research objectives directed at Fragile X. The first is we're trying to develop human stem cells as a way to study Fragile X. So, for example, some of our recent work is geared towards taking advantage of a recent development where you can take skin cells from an individual and turn them into stem cells, and then those stem cells can be further turned into brain cells. So we're hoping in this way to be able to make Fragile X specific stem cells from individuals who have Fragile X. So that's one area. The other thing we're doing in my lab is to use these human stem cell models that we are developing to ask very specific questions about what goes wrong during brain development in Fragile X. So, for example, over the last few years, we have found a signal that is not working correctly in Fragile X brain cells. And because this signal doesn't work, it impedes the way these brain cells communicate with each other. And so we've just recently found this, and so we're spending a lot of time trying to figure out how does this happen, what kind of symptoms may it cause in people, and potentially can we find a way to target this signal for therapeutics. Many researchers who are working on Fragile X are very interested in drug therapies, because many of the symptoms of Fragile X are amenable or open to different kinds of drugs, at least treating the symptoms. And so I think that many researchers are trying to are doing more testing of drugs than researchers in studying other developmental disorders. And I think that, at least from our perspective, if we have human cells that we can test specific drugs on, we can bridge the gap between animal testing that's done at pharmaceutical companies and clinical trials going straight into people. It would be nice to know before you try drugs on people whether they do the same thing on human cells as they do on animal cells, for example.